All right, looks like we got about 30 people in registrants and attendance. We had about a little over 80. I don't know if if you caught the most recent number. I know it was north of 80 this morning that have registered. This recording will be available to anybody that does not make it here for the live event, but there is some features that are only available to people live. The polling, the ability to respond to some of the presenters is only available live, but if you're doing this on your lunch break and you end up having to leave, we will get a recording out to everybody that registered, and you'll be able to see it there as well as on YouTube. All of our previous recordings of the Petri Dish are all available through Big Marker, so if you missed any of our prior sessions on direct-to-consumer testing or ECRA compliance, any of that, you can find it there. We've done one on the lab shortage most recently, had almost 400 people in attendance there, just going over some of the market statistics of what's happening in the lab industry and how many people are retiring and coming into the field, and what that might mean for your lab. But today we're going to focus on liquid biopsy. It's one of those things that's really exciting. People hear about it, not quite sure what it means, not sure how to get involved in it. Is it a good thing or a bad thing for us as lab owners? And so we're going to go over that today and just talk about liquid biopsy and give you a little bit more information. Before we do that, just giving you a soft pitch for our services. We do this for free. We don't charge the presenters anything. We don't charge the attendees anything. We do this in order to foster a more healthy lab environment, because we think lab is underutilized, and we'd love to see the lab, the image of the lab lifted inside the community. 75% of medical decisions are made based on laboratory testing. Less than 3% of the money goes to the lab, right? So doing a lot of the work, driving a lot of the knowledge that is driving healthcare, not seeing a lot of the recognition that goes with that. I think a lot of the movement right now is towards getting that data, those medical decision information, to the patient in a more efficient manner through whether it's direct-to-consumer testing, offering them some of this general screen that might be possible through liquid biopsy. There's a lot of really great innovation that's happening right now. We're excited to be a part of it. What we do, we don't own labs. We staff labs. We build labs. We help buy and sell labs. So if you need any of those services, we hope you'll keep us in mind. We do run the largest communities of medical lab scientists in the world on LinkedIn and on Facebook. And we provide the medical lab directors, our directors, direct about 170 high complexity CLIA labs around the country. So we like to be a one-stop shop for lab owner and lab needs. So if you have any pain points that we can help you with, we hope you'll reach out. And then as you grow, we'd love to help you scale. So that's enough of my pitch. I want to get into what is liquid biopsy. So we have some polls going now asking if people think that this is what it's cracked up to be. Just big picture, because I know our presenters are going to jump right into deep science. And so I wanted to give you kind of an overview of what liquid biopsy is. I threw the definition of it out there for you to read. I won't read it word for word, but basically we're looking at a difference from taking a standard tissue biopsy to doing it through liquid, right? So there's a lot of benefits for that, not only in comfort of the patient, but being able to possibly detect things that a patient isn't even expressing, right? So they might be able to be screened for some of these cancers that they normally wouldn't unless they had some kind of symptoms that had presented. There's just so many possibilities that are freed up by that. Normally, these liquid biopsy tests are working through circulating free tumor DNA, cfDNA. You'll hear that term quite a bit throughout the presentations today. And liquid biopsy, traditionally people think blood, but we do have also a urine presenter today. So we have 3 of these technologies are going to work based on blood samples. One is a urine sample as well, but any bodily fluid or liquid would qualify under the umbrella, at least in my opinion. And so it is a little bit loose in terms of definition, but I think that all these presenters fall under that umbrella nicely. Becoming the new standard of care, I threw a quote up there from Dr. Solit just on what does liquid biopsy mean. I'm really excited most about how it could become a standard of care, that it could be used as a screening device, and that it could save a lot of lives through doing that. And so that's the goal. That's what I would like to see happen, and I think it's just better healthcare. So We'll go ahead and introduce our first presenter. His name is Oded Byron. Oded has over 10 years of experience in life science industry, majority of which are running various aspects of clinical lab companies. Oded has a track record of leading the ops, finance, business strategy for publicly traded high-end molecular diagnostic companies. He's now moved on to take on the co-founding role with Dr. Erez Yitan of Neurodex. So they're a non-invasive neurological diagnostic liquid biopsy. And Oded, I will go ahead and turn over the reins to you so you can go ahead and take control of the slide deck and tell us a little bit about Neurodex.
So thank you. First of all, I just wanted to address two issues. One, Neurodex does not deal with cell-free DNA. And two, we also believe that liquid biopsy is not limited necessarily to the realm of cancers. And this is part of what we will demonstrate in this upcoming presentation. So the problems that we intend to address are bringing effective non-invasive diagnostics for brain-related conditions into the neurodegenerative space. We believe that these could assist in increasing success rate of clinical trials in a field that is usually considered as a graveyard for therapeutics. Alzheimer's, Parkinson's, ALS, traumatic brain injuries, frontal temporal dementia, and others, all of these are fields and diseases and conditions in which there have been numerous failures in clinical trials. If you probably all know about the recent revival after the demise of Biogen's attempt at Alzheimer's. We know that a lot of dementia patients are misdiagnosed, and if you look at treatment for psychiatric diseases, a lot of it is trial and error. And so we believe we have the technology to address that. The technology we're utilizing is called exosomes, specifically neuronal exosomes. Just a little primer, exosomes are unique extracellular vesicles that are secreted by cell types into the blood, and their payload reflects their cell of origin. We utilize a proprietary immunoaffinity-based isolation method, and we attempt to provide answers in advanced drug development utilizing this analysis. Give me a second, next slide. So we attempt to assist in all stages of therapeutic development, from target identification and discovery through target validation, preclinical and clinical, through cost-effective analysis of differential expression in human brains. We can also check for target engagement in animal models and later in human models. We want to validate target expression in the human brain, and in the clinical trial setting, we can assist in patient selection and stratification and discovery of endpoint biomarkers. In addition to assisting pharmaceutical companies and therapeutic companies in their development processes, we also have the ability to become a platform diagnostics company for various neurodegenerative indications. Just as an example, and I'm moving to the next slide, we have here an example of neuronal-derived exosomes utilization in literature. One of our co-founders, Dr. Yitan, had successfully published a study that showed early detection of Alzheimer's disease with 83% specificity and 91% sensitivity. And we have developed a protocol for increased accuracy than that one in terms of specificity, quality control, process, and robustness. And moving to the next slide, sorry. In the last year, we have amassed a lot of data validating and proving that our technology works. As I previously stated, we have 85% sensitivity and 90% specificity in detection of Alzheimer's in patients with mild cognitive impairment. We have also completed a small proof of concept study for pathology stratification in frontal temporal dementia. We have validated, optimized, and quality controlled our NDE isolation and our spiking system utilizing fluorescent labeled NDEs. And we have consistently and specifically captured neuron-derived exosomes and proven that this methodology is compatible with proteomic and lipidomic downstream analysis, as well as utilizing different detection technologies, including Luminex, ELISA, and MSD, and others. In addition, we were awarded an NIA SBIR grant recently, and to expand on our validation and our in-house proof of concepts for Alzheimer's disease, we had a completely blinded study of a cohort of 20 individuals with mild cognitive impairment and 20 cognitive normal controlled, which we analyzed. We utilized a 4-protein algorithm comprised of 4 proteins that are found within neuronal derived exosomes as part of their payload, and achieved the specificity and sensitivity described above. We also, as a sidebar, did a small cohort of 9 frontal temporal dementia, in which we had successfully been able to stratify the 2 major pathologies for frontal temporal dementia. And moving on. In addition, another validation step, we basically took mice of various Alzheimer models, as well as healthy control mice, and analyzed their plasma and NDEs in their plasma, and compared it to the cortex after sacrificing the mice. And we have showed compatibility between the mice plasma and their cortex for multiple proteins, including tau, phospho-tau, and others. This proves, in our mind, that there is a potential for utilization in therapeutic engagement, and as well as proving that the payload of exosomes actually does show the cell of origin status at the time of their analysis. Our advantage is that we are able to use frozen samples, and so we're enabled to provide our clients with retrospective studies and utilize external biobanks. We are able to identify diverse pathological aspects in neurons utilizing our capturing technology. We can combine our isolation platform with different methodologies, and our platform is robust and highly precise, as well as proprietary. Now, while we are neurology focused in our research and development, we've also found some methodologies for isolations of other cell types as exosomes, which we may be interested in either out-licensing, partnering with, maybe doing a joint venture development with potential collaborators. Our team is comprised of industry veterans and scientific bright minds. Dr. Eitan, who was the first scientist who researched exosomes for neuronal diagnostics in the NIA, has over 27 peer-reviewed papers and 18 of them on NDEs. Myself, as John stated, I have been in the diagnostics area for a while now, in public and private, raised over $60 million through public and private means, and managed various aspects of diagnostics companies and life sciences companies. And our controller, Christina, who has been both in high tech and life sciences and has a track record of raising over $50 million in her various roles in these companies. We currently have some commercial collaborations which are confidential, as well as academic collaborations with the likes of the NIA and the NIH, Columbia University, MGH, as well as international collaborations with Tel Aviv Medical Center, as well as a sub-unit of the U.S. military. And we are delighted for this opportunity, and we thank you for your time and putting up with us. Thank you.
Thank you very much, Oded. That's great. That's pretty exciting and, you know, fascinating. And I think that's going to be game changing. And so I just put out there a promotion for you guys to click on if you're interested. So if you have a lab, would like to get into the liquid biopsy space, this test sounds like it would be something that'd be interesting to some of your customers. Go ahead and click on that, and we'll make sure we make a connection between yourself and Oded. We're not looking to get anything out of that. You'll see his information is on the screen as well. If you wanted to reach out to him directly, that's also fine. We're just trying to foster some connections here. And I see that in the polls, looking through those, that 65% of attendees are saying that they would like to add liquid biopsy to their lab's test menu, but they don't really know how to do it. So this is an easy way to go ahead and do that and execute on it without having to do the R&D piece. So a good potential partner. All right, and so if you have any questions for Oded, you can chat those over to us in the chat function. That's on the right side of your screen. You'll see chat, Q&A, and polls. So feel free to ask any questions you have there. I don't see any that have come in yet, but you can still ask those and we can address them at the end, or we'll connect to you after this. But we'll go ahead and we'll move along because I know we have several presenters here and we want to be respectful of everybody's time. So we're going to have James Howard Tripp of Stage Zero Life Science up next. He's the chairman and CEO over there. Stage Zero Life Science dedicated to the early detection of cancer and multiple disease states through whole blood. Aristotle, the company's new next generation test, is a panel of simultaneously screening for 10 cancers from one single sample of blood with high sensitivity and specificity for each cancer. Aristotle is built on the company's proven and proprietary Sentinel principal technology platform, which has been validated on 10,000 patients and used to develop the first liquid biopsy for colorectal cancer, which is called colon Sentry. In addition to building a pipeline of products for early cancer detection, the company operates a CAP-accredited and CLIA-certified reference lab in Richmond, Virginia. That's where they offer that colon Sentry test, as well as some licensed biomarker tests for breast and prostate. So with no further ado, Jim, if you're ready, we'll go ahead and hand over the deck to you so you can go ahead and drive.
Marvelous. Thanks, John, and good afternoon, everyone. John, can I ask you to advance the slides for me, please?
Absolutely.
Okay, so
go ahead and we'll move.
Stage Zero Life Sciences named for that because our focus is particularly on finding disease at its earliest stages. And is some earliest stage of disease is being known as Stage Zero. That's actually how we've named the company. We're one of the very early pioneers of liquid biopsy. We've been at it since 2001. For the first 10 years, we were essentially in the wilderness. No one believed we could do what we said we could. So it took a fair amount of time to get it through. After about 10 years, we started to be accepted. The following 5 years, it started to become absolutely mainstream, and now it's quite normal. We're different, I think, we certainly don't do circulating tumor cells or cell-free DNA either. Most of that, I think, we would regard as being post-diagnosis. We're all about pre-diagnosis. We're all about finding it very early, and so we'll go from there. The issue is, and staying with the slide that's on the screen, is that most cancers these days are diagnosed late. When they diagnose late, your survival rates are low, your costs are high in terms of treatment, and we're all about trying to solve that. I will also say that we're not just in cancer. Next slide, please. A science, which we call the Sentinel principle, is based on the fact that circulating blood reflects in a detectable way what is occurring throughout the body. So our system is dynamic. We can do this multiple times a day. We can do it multiple times a week. We can do it multiple times a year. And each time it will actually give you a reflection of what is actually occurring in the body at that particular time. It's based on immunoediting and the fact that subtle changes that occur in cells due to injury or disease trigger detectable changes in gene expression. These we measure by mRNA gene expression, and what we do is we narrow down. We typically always start with a large number of genes. We will then narrow it down to where we're actually getting the best disease signal. We will cross-check the relevance of the genes, and from there we will go ahead and develop the product. We have named and patented this technology. It's the Sentinel principle, and you can find those patents out there. So we did the first liquid biopsy for colorectal cancer. We named it Colon Sentry, and we did the first ever 10,000 patient study in North America. We were actually the group that showed that the point prevalence of colorectal cancer is 0.7%. We followed this up with validation in Asia. It's now been utilized in over 100,000 patients in the U.S. It detects some colorectal cancer in stages 1 to 4, but is very strongly biased towards stage 1 and 2. We will also find polyps, and I'll talk a little more about this, particularly in our follow-up program. We also can see right-sided lesions equally as well as left, and so why is that important? Well, it's important because colonoscopies miss about half of the right-sided lesions. Stool tests, according to the latest data, seem to miss the vast majority of right-sided lesions. Yet right-sided lesions are the most aggressive and the most deadly, so you must find them. You will find that if you use our test, or at least the data shows if you use our test, in advance of moving on to a colonoscopy, you will find 2.1 to 4.7 times more lesions. And so it's very, very effective in terms of this. We're also very clear that the true diagnostic in all of this is always the piece of tissue in front of the pathologist. So everything that we will do is to give the probability, the likelihood of you having the particular issue, in this case colorectal cancer. You would always then get referred in for colonoscopy if you have an elevated result, if you have a raised probability, and it is obviously the biopsies we say. We think this will change over time, but certainly the state of medicine is that today. Can we move to the next slide? We started with a program called Aristotle. And so Aristotle, we started in osteoarthritis and rheumatoid arthritis. We found, for example, that we could see those diseases in very young adults. We could actually see it progress. We could see the onset of when it became symptomatic. We were working with Pfizer and Eli Lilly back at that point in terms of looking at the various treatment modalities and being able to actually help select which of the treatment modalities would be most effective. Both Pfizer and Eli Lilly decided not to go forward with their RA and OA programs, and we moved on. We moved to neuroscience, and so it was interesting to see where you were going. Neuroscience, I think, is a very fascinating area, and you're absolutely correct. There's a lot of need there. We focused into cancer, as I think is an area many people often go to. We went to colorectal cancer first, and from there we've now expanded out, back out to the full Aristotle program, but into a multi-cancer platform. And at this point, off of a single sample of blood, we are looking for 10 specific cancers, and I'll talk to that in a fair amount of detail. We have high sensitivity and specificity. And key with this, we can fully, can you go back one? There we go. We can fully discriminate each cancer, and that's really critical. The other thing, too, is this is not research. It's proven, it's done on a proprietary technology platform that has given rise to Colon Sentry, and Colon Sentry, as we said, has already been used in 100,000 patients. Next slide. The sensitivity and specificity here is the result of about 3,000 patients at this point. We are beginning to add patients to this. We will ultimately probably look for about 8,000 patients. That's not because we believe you have to have the magical 10,000, but I think it continues to build out. Where Colon Sentry is an LTD with New York State approval. In Aristotle's case, we will begin as an LTD, but we will then almost certainly take it forward for FDA approval. And that is because of what it is. Next slide. So why are we different? And I think this is critical. A lot of the tests, and particularly if you go to cell-free DNA or circulating tumor cells, because a lot of this is still focused on the work done post-diagnosis, and people are trying to pull it forward into the early diagnosis. One of the big issues as yet is that you might get an indication that there is cancer, but sorting out exactly which cancer it is not immediately evident. And so one group that we're talking with on Aristotle right now, where we had approach from them, they said, well, when we use some of the technology, we may get a flag that there is something there, but we're now into a 6 to 12 month, $12,000 to $50,000 search to try and sort out whether, one, is it cancer, and then two, if it is cancer, which kind of cancer. And so this is why we're very different. Because we're doing the 10 cancers together, we're doing it off of a proven technology platform. At this point, we can fully discriminate and detect each cancer with high sensitivity and specificity. 10 cancers is what we're taking in to begin with, but we obviously have the opportunity to add to this. It's simply a matter of doing the work. We've also split them into male and female panel. We should be in the market as a start sometime this year. We, in actual fact, have just agreed on our first collaboration, but we will continue to build it out and, as I said, ultimately go for FDA approval as well. Next slide. So we're particularly pleased about the panel for women. Why? If you have a look at the sort of female-specific cancers, if you know anyone who's had ovarian cancer, it's an absolutely miserable disease. And so if we can do something about finding it early and finding it with great reliability, i.e. high sensitivity and specificity, we think that is huge. Obviously, cervical, endometrial, and then getting to breast is important too. We currently have a breast cancer test, but it augments the mammograms. It also is specific or has greatest utility, I think, in women that have dense breast tissue, so that's about 50% of women. This would be the next step up on that. And so we will take this in. Move to the male panel. Next slide. Male panel, 6 specific cancers. We're looking to add lung to all of this as the next step as we go through. But again, that will begin to move through. This is a step up from colon sentry, for example, in that the colorectal cancer test we have there. This will be expanded as we've learned things as we move forward. We will have a lot more identification of polyps in terms of where it is, and I think, as we all know, actually detecting and removing polyps is one of the greatest effects that we can have on preventing colorectal cancer. Next slide. Our market opportunity is a little different. We deal with physician practices. That, I think, is always an important part of this. But I think based specifically upon where Aristotle is going, so it's not only the current test, but also where Aristotle is going, we've taken a lot of time to build out a very strong telehealth program. This is very specific to patient-directed testing, but it also is a critical piece as it's moved into working with large health systems. They, too, need the telehealth aspect, and very particularly when you work with employers. So right now we're dealing a lot with large healthcare systems, and by that we would mean everything from the large insurer that would invariably own the large healthcare system, down through the hospitals, the clinics, and then into their outreach program. Outreach often can only effectively work because of telehealth. And then we're also working with employers, particularly large employers, with all of the benefit that drives back across this. So our approach has been in all of that, and the thing that we can tell you about this particular approach is that there is very good acceptance. We're also, although we've been very heavily focused into U.S. Commercialization, we're also beginning to move out into Europe and Canada. And so to finish off with a little bit of information about us, as I mentioned, it's really nice after having fought the battle for so long to actually have our approach actually controlled. Sorry, back a slide, if you will. There we are. We operate a CLIA and CAP certified lab in Richmond, Virginia, and I think, as we mentioned, we've done over 300,000 tests. And although we currently offer the 4 tests, that you would have lung included in that, Aristotle is the one that we truly are most excited about. And so what would our interest be? Obviously, working with people, with partners in the space. We think that that's the way you actually achieve success. And as an organization that's beginning to move very strongly into its commercialization phase, there's always a need for money. So investments are very welcome. John, that's it.
Thank you very much, Jim. I appreciate it. Also amazing, and it's great to hear what you've been able to do and to see your vision for doing that really early on and kind of breaking the soil for a lot of other groups to get into this space. So we appreciate the work you've done there. All right, if you have any questions for Jim, feel free to drop those in, and we'll get towards those at the end. Next, I'm going to, just in consideration of time, we're going to move quickly over to Sam Solomon of Mir Scientific. Sam's a serial entrepreneur, renowned executive throughout his career, senior executive at board-level positions, developing game-changing technologies in companies across industries, including biotech, art authentication, aquaculture, financial services. He's a jack of all trades. And Sam attended the American University of Paris in France, and then also Malcaster College in St. Paul, Minnesota. He's a member of the Board of Trustees and Board of Selectors at the Jefferson Awards for Public Service. I first met Sam at the World Trade Center towers, where they have their corporate offices, and we were scouting out office space in a skyscraper in Manhattan. And I said, that's a really expensive place to build a lab, but they had some good reasons for doing so. And so we're going to go ahead and turn the deck over to Sam so he can tell us a little bit about what Mir Scientific's been able to accomplish lately.
Thank you so much, and nice to meet everyone, and great job on all the presentations today. I just want to take a quick moment to shout out to our friends at Lighthouse. They're a formidable company. They're a great partner, and for anyone contemplating on using them, we have had nothing but the best of experiences. So please don't hold that against us, but this is we thought it was important just to share that. So thank you very much. As our colleague said, we are in the business of solving what we consider to be significant challenges facing humanity in our group. One of these, of course, is the concept of screen-level diseases, particularly around cancer. We've developed a platform that is focused mostly on different types of non-coding RNA, and our goal is to be able to provide what we consider to be the profound possibility of liquid biopsies in molecular science, and that is to create precision at both the individual patient level as well as applicability at the population level. And so, as we know, many of the tools of bioinformatics prevent, when you measure NPV and PPV, their case versus controls. When they're applied at the population level, their performance, obviously from a mathematical point of view, diminishes. And it's very hard to have a tool that is both as effective as it is at the population level, meaning, for example, age-eligible men, or as it may be in other cancers, men and women, as it is for the individual. So please keep that in mind. We view ourselves as differentiated significantly for that reason, and we were happy to share with you what we've accomplished. So this is one aspect of our platform. We've focused on prostate cancer because it is a broken standard of care. The clinical pathways suffer from a series of cascade challenges, mostly because the initial screening tools, which are recommended by the government and, in fact, by most countries in the world, begin with the PSA. It is a blood-based test, and the PSA is a test that effectively is better than nothing, one can say, but not that much better, because it misses early-stage cancers that present below an arbitrary level that's selected as a cutoff, namely, for example, 3 or 4, which, depending on which geography you're in, are the cutoffs. And it also has a very high false positive issue. Those people with false positives end up having to have unnecessary interventions. These interventions are both cumbersome, expensive, and they consume significant resources in the form of the clinical pathway. So a biopsy is not just the doctor performing the biopsy, it's the histopathology that's required, it is the anesthesia that's required, and unfortunately, there are morbidity and other complications that have a significant enough impact so as to affect both quality of life and the DALY in terms of daily active productivity of a person undergoing those. So we, as mentioned, we do in fact interrogate different types of non-coding RNA, and we do so through a process that is based on urinary exosomes. The exosomes that have been described by colleagues presenting earlier are mediated through different types of cells. We believe that exosomes in general represent a new class of biology in terms of intracellular communication, so they're significant. We do not interrogate the exosomes as a function of the exosome composition. Our goal is to retrieve from these exosomes the encapsulated non-coding RNA entities that have been captured within the cells and excreted. Because we're dealing with a system that primarily excretes into semen and into urine first, not into serum, we viewed the urinary approach as the most non-invasive and scalable approach. However, we initially tested directly the same process to interrogate non-coding RNAs through biopsy materials as well. So we call ourselves a Sentinel. Sentinel is a popular name, we gather, and it is a Sentinel test because it's an early indicator of issues. And we have developed not a liquid biopsy test, we have developed a platform within which is included different types of tests to track the continuum of disease. And what that effectively means is that most cancers are dynamic, they change in status, and it is vital not just to be correct in the initial diagnoses, but then to be able to characterize the different types of questions that have to be answered to create the optimal clinical pathway for the patient. So in this case, what we have been able to do is first assess whether there is cancer or not. And just to be clear, this is a simple urine collection. It does not require any pre-preparation of the patient. There's no special regimen that's required prior to the collection of the urine. The urine is simply collected. It is cooled and sent to our labs. Our labs, which will soon be CLEP and CLIA certified to be able to deliver the test. So right now I'm discussing this at the research and validation level. The lab is able to then perform the test in a high throughput environment. It takes around 3 business days for us to produce the results. And in essence, we can tell the following pieces of information from one urine specimen. For those who are in general, let's just say the affected population of, for example, men between 45 and 80, we can say definitively whether they have cancer or not. Now, when we say definitively, we believe that we are far more accurate than the gold standard, which is biopsy, because biopsy has a highly observed false negative rate. If you think about biopsy, it's a kinetic process, and you're trying to target physically a tumor, which may or may not be within a convenient area of the biopsy, including MRI-guided biopsies, because they all have certain types of false positive and false negative, particularly the negative limitations. And depending on the urology groups and others, it's somewhere between 15 to 30% at times. So our test observes the characteristic from the entire gland, and not just a section of the gland, or not just a specific condition of the gland induced because of a massage or some other type of intervention to produce the specimen. And so we are able to tell from that urine, is there cancer or not, which we call our PCA test, or prostate cancer test. We then can further determine from the same urine, for those of that cohort who have prostate cancer, we can then determine the clinical pathway as to what is it that they have. And in this case, it would be either a form of cancer that needs to be surveyed. It's called something called active surveillance, which means that they do have cancer, but it's unlikely to be progressive disease and can be simply observed. If in the course of observation, there is a tendency for the status of the cancer to change and become worse, it's called, effectively, it's called progressive disease. At that point, the patient can then go through an interventional standard of care, which would then include a treatment paradigm such as biopsy, fusion by MRI, etc. We can also therefore tell from that same specimen whether that cancer is of the class of cancer that requires immediate intervention. In other words, it's clinically significant. Because we can tell these things from a urine specimen as a indication of that current patient's disease status, we can then serially follow the patient as they go on a protocol for surveillance. So we can then not only determine their status, we can continue to monitor their status as is to see if there is any difference in their effectively what we call our microRNA and non-coding RNA signatures, which reflect a change in the status of the disease. So this is in essence the differentiation that we believe the standard of care and the population requires is one in which the continuum of care follows the patient from different modalities of treatment, so from primary care to specialist care to post-surveillance, in other words, post-determination care, and it does so through a single platform with very high accuracy, and we'll describe the accuracy. One of the reasons we're able to do this is that we started with a view, even though we have several leading molecular scientists and particularly cancer genomics experts, including our co-founder and chief scientist, Dr. Martin Tenniswood. His approach was to, of course, assume that we do not know which specific biomarkers are informative, and that we should first begin by surveying the entire available types of sequences, in this case, non-coding RNAs. So these are not messenger RNAs. These are smaller species of RNA that have specific different types of functions than effectively coding and other expression functions in the DNA functionality of cells. So from our point of view, we developed a statistical methodology and a classification system to interrogate the large possibilities of disease and the sequences that are involved there. We selected the ones that are most informative, which are a mix of non-coding RNAs, microRNAs, and small nuclear RNAs for each of those dichotomous groups that I just mentioned. So cancer, no cancer, the type of cancer from the point of view of aggressiveness and the likelihood of progressive disease. We then optimized those on a proprietary assay, which uses a system that Thermo Fisher produces as what's called a Quant Studio system, and it's a high throughput system because it's not a full screen at that point, a full genomics. And so you can see here the Quant Studio as well as the liquid handling system. This is what the assay looks like. We can process several thousand patients a week at the minimum, and we're scaling up to be able to produce 1 million tests in our lab per year. So the performance of the test against the highly tracked cohort representing a diverse patient population produces a high sensitivity and specificity for the PCA test, which differentiates between cancer and no cancer. So here you can see some of the cohort. The sensitivity and specificity are in the high 90s, and as are the positive and predicted negative values. The PPV and NPV to us is of less importance at the population level than it is at the patient level. So when you're comparing cases to controls, PPV and NPV are interesting numbers, but the real numbers that matter are the sensitivity and specificity, because they're not just simply a mathematical function of comparing what happens to be the selected number of cases versus the selected number of controls. So this is a population level number, and it's very important to us. As Odette had mentioned earlier, sensitivity and specificity are very important determinants of the efficacy of the platform. So I'll just zoom in. If you can kindly see here, this is the disease management platform in terms of its capabilities. From a single urine test run for a single patient, we can determine whether the patient is in fact one that needs to be in a cohort that has a type of cancer, and one that is not and can go home and come back for a later screening whenever their annual checkups are, etc. For those that do have cancer, the test is able to determine, the platform is able to determine the virulence of the cancer. So in this case, we're looking at what is the equivalent of grade group 1. Grade group 1 is viewed as a favorable or low-risk cancer, and grade group 2 through 5, which are viewed as clinically significant cancers. And that group comprises of, this is based on a Gleason system. It's observed by looking at histopathology from either biopsy or radical prostatectomies. And so this is effectively compared to them. The challenge that we have as, for example, other types of diseases would have, is that the gold standard, in this case, biopsy, is relatively imprecise because of its false negative rate. So we're mindful that we also had to look at patients who then progressed sufficiently enough to have had their entire prostate removed and had to go through a what we call a radical prostatectomy. So we've compared our results to both. We also have a platform for bladder cancer that's based largely on the same approach. The sequences involved in bladder cancer are different than the ones in prostate cancer, but were derived and determined through the same methodology of looking at things in an unbiased way using our proprietary statistical classification and eventually the prediction systems that are used to dichotomize the patient specimens into those cohorts. The purpose of bladder cancer screen is to eliminate from cystoscopy the patients who present with risk factors that are common. And in particular, these include something called gross hematuria and microhematuria, effectively blood in someone's urine. And this applies to both men and women. And we have a very, very high accuracy, both in the high 90s for specificity and sensitivity, and so therefore predictive values are equally high. So thank you so much. And we just as a quick status as to where our company is, we are working in different geographies. We're active in North America, both in the United States and Canada. We're active in Israel and the EU, and we are developing platforms for Asia. So we're very much of a global company. We're fully funded, and we're about to launch our platform for commercial use under an LDT through both the CLEP and CLIA provisions of basically an LDT operational purview. Thank you.
Thank you, Sam. Make a very good case for why liquid biopsy should maybe be the new gold standard instead of tissue. Appreciate everything you guys are doing to just break down those walls and increase adoption and awareness for these types of testing. We're going to move over to John Cucci of Accupath Laboratories. John has been in the anatomic pathology industry for the last 17 years, serving as an executive in different leadership positions, mostly focusing on sales, marketing, and strategy. John has a particular interest in leveraging mutually beneficial lab-to-lab partnerships. He's been around for a long time, knows a lot of labs, is open to collaboration, and so I encourage you guys to check out his new innovative discoveries and figure out how you can partner with him to leverage that at your lab. So I'll go ahead and I'll turn it over to you, John, if you're ready.
Great. Thanks, John. Appreciate the time here, and I will jump right into it. Appreciate that intro and a pretty good segue there from the last presenter mentioning bladder cancer, because we'll segue right into that at this point. John just gave a quick bio about me, so I can skip over that. But what we'll do today is, I want to introduce you to a new bladder cancer biomarker that we launched last year at Accupath Laboratories. It's an innovative new non-diagnostic screening tool for the detection of bladder cancer. That's not only clinically significant, but also can help generate much needed revenue for your laboratory. So it accomplishes, two of those very important, buckets: clinically significant and, a revenue generator. Just a quick slide about Accupath. We've been around for 20-something years, based in Plainview, Long Island. We're a multi-specialty anatomic pathology lab with urology and GI representing two of our focus specialties, also in derm, hemo, podiatry, and GYN. We do service clients nationwide. We're in network. We offer services both globally, where Accupath renders the diagnosis, and TC only, where our physician customers and or our commercial laboratory partners can retain the professional component if they wanted to engage in that model. So, with client pricing available. We do have subspecialized pathologists, and our chief medical officer is Dr. Hillel Kahani, who for the last 30 years or so has been a leader in the GU pathology space as executive medical director of both Bostwick Laboratories most recently, and prior to that, Dianon Pathology. Accupath offers an extensive fish menu, as you see there, highlighted, in this case by Eurovision. We have a Barrett's esophagus fish test, P10 ERG for the detection of for prognostic marker for prostate cancer, and some other markers as well. Skip ahead here. So bladder cancer, just quick overview about why, what the state of the market. There's 80,000 new cases annually, mostly men, but women as well. The sixth most common cancer in the United States and the most expensive to treat, and that's due primarily to the very high recurrence rate. So that's what we're looking to attack here with our new marker. 6.7 million people with hematuria, 700,000 people with bladder cancer, and as everyone is probably aware, the leading risk factor for bladder cancer, certainly smoking is the number one risk factor with a 3x risk factor there. Chemical exposure, tainted water, family history, first responder. These are all important for the background of bladder cancer. So with regards to bladder cancer screening, John, are you advancing my slides? Just want to.
Sorry, I was on mute, but no, I am not. So if anybody else is messing with the controls, just hands off a little bit. We'll let you drive, John, but it's not me.
Yeah, thank you. They're trying to move me along, but my slides are advancing. So yeah, somebody could just back off on that. With regards to bladder cancer screening, the current gold standard, utilized by urologists, is a cystoscopy. However, cystoscopy, while being sensitive and specific, it's invasive. It does cost the system quite a bit of money and is also, less effective with flat tumors. Urine cytology is the traditional non-invasive screening tool that's utilized. The issue with urine cytology is that it has a very low sensitivity, especially with regards to and its inability to pick up low-grade tumors. So, while it has a high specificity, when it does, pick up cancer, that's fantastic, but it does miss an awful lot of cancers out there, and so that's why it's known that there's a significant unmet need in that marketplace. Eurovision is available as well. It's been FDA cleared for the last almost 20 years. Pretty good sensitivity, lower specificity, but it's also very expensive, and it also does not have the ability to diagnose or pick up low-grade tumors. So there's, urologists are ordering less and less of these tests because of those issues. And so, there's a need for, something new out there. And our solution for that is a test that we are branded Euro 17. We have, keratin 17 is what our test, identifies in the urine specimen. It's a simple voided urine specimen that's provided. And in the work that Dr. Ken Schroyer, who's chairman of pathology at Stony Brook University, here in New York, and his co-founders, see keratin 17 expression exists in a wide range of cancers, as you can see from the chart, but it's especially high in bladder cancer. So, due to the poor screening methods that are currently out there right now from a non-invasive perspective, and for the due to the large market size of patients who have bladder cancer and recurrence. The team has identified bladder cancer as the first cancer to where to employ this marker. There have been several published papers about the sensitivity and specificity and just overall clinical utility of Euro 17 in this space. There are additional studies being published this quarter, including several from Europe. Back in November of 2019, the FDA granted Euro 17, a breakthrough therapy designation, which is going to fast track a clinical trial, which will be kicking off this quarter across the United States, leveraging several of AccuPath's customers as client sites. So Euro 17 is an immunocytochemistry. It's billed as an 88342. So every laboratory that's likely in this space is familiar with the 88342 code, which is paid for by all payers. So they're, at least for an AccuPath experience, we have not had any issues with reimbursement. The published studies have shown high 90s on both sensitivity and specificity. And the beauty of Euro 17 is that it's, it's specifically highlighted in patients that have urothelial carcinoma. And often, this marker is picking up changes, or the keratin expression prior to anything presenting morphologically for the pathologist, or visually for the urologist. So just like Eurovision's anticipatory positive designation or disclaimer per that test, this can often pick up changes that are happening behind the scenes and identify patients who are at very high risk of either developing bladder cancer or have already developed bladder cancer. Currently, Uro 17 is being used in conjunction with urine cytology. So our pathologists at AccuPath and our pathologist clients in the marketplace are using the Uro 17 slide in conjunction with their traditional ThinPrep slide, while also utilizing, Eurovision FISH, perhaps if Uro 17 is positive. And we also do have some customers who are using and leveraging Uro 17 as a standalone test. They may do some of this testing, and this may be the case for your laboratories as well. You may do, this in-house. You may do cytology in-house, but want to, view this marker in conjunction with your in-house slide. So we do have, many clients that just order just the Uro 17 marker from us and, do the rest in-house. AccuPath has a semi-exclusive arrangement with, the company that manufactures the antibody, and but we're currently, offering that nationwide. From a pathologist perspective, there have been other markers, IHC markers in recent years in this space that have not been met with as, as positively, specifically due to the difficulty in interpreting it. As you can see here from these slides here, it's very clear whether this marker stains or doesn't stain. If it does stain the urothelial slide, that indicates a positive case. If it does not, that is, a benign or a negative case. So it's very, straightforward for pathologists to interpret. There's not a lot of, training required, and that has certainly received a very valuable feedback. So from a clinical utility perspective, I'll just run through this quickly. So with regards to, traditionally, if you get a urine cytology result that is benign, it's very difficult to know, is that a true benign because of the low sensitivity. But a benign cytology with a negative Uro 17 case, and due to the test's high negative predictive value, that is a confirmed negative case that, pathologists and urologists have found very helpful. Conversely, a benign cytology case that's otherwise unreliable with a positive Uro 17 marker, indicates a patient that does need, additional workup and close follow-up. Perhaps a reflex to Eurovision, certainly a cystoscopy and a biopsy. Same thing for the atypical cases. So what it really does is it's a risk stratification tool used in conjunction with current methods out there to put those patients either into a lower risk bucket where we are, very confident that it is not bladder cancer, perhaps causing their hematuria or other symptoms that they're experiencing, or conversely put them into that higher risk, close monitoring bucket where that patient needs to be seen, needs a cystoscopy, it needs to be seen quicker. So the whole goal is to, rule in the proper test when necessary, and certainly help patients financially and, clinically and physically avoid, unnecessary tests and treatment that they do not need. We've been out marketing this test for the last, 6 to 8 months. We're partnered with several of the largest urology groups out there in the country. You may be familiar with the term LUGPA, the biggest groups out there that have in-house laboratories and always interested in new tests. And it's been received very, very well from our client base. The Bladder Cancer Advocacy Network and down in Washington D.C. recently met with us and is going to help spread the word as well. Very briefly, of course, urology is a target market, but also the GYN market is a big market as well, and that was highlighted by the Bladder Cancer Advocacy Network. Many women go undiagnosed when they visit their gynecologist, perhaps with hematuria or blood in their urine. Primary care, internal medicine, the front lines as a screening tool. I mean, as a simple voided urine collection, it's quite easy to collect specimens from those patients and screen more patients that likely right now are not getting properly screened. And most recently, I was made aware of some studies, with regards to opioid users and, the high prevalence or the higher risk of having bladder cancer. So this is a newer market, something we're looking at, not a space we're in right now, but, worth mentioning that there may be an opportunity to identify patients in that market as well that are at high risk for bladder cancer. So just in, a summary, we partner with, hospital-based laboratories, pathology practices, commercial laboratories, or independent practices. We're open to partnering any kind of way. We can facilitate the purchase of the Uro 17 test if that is something your laboratory is interested in. or like I said previously, we can engage in a TC/PC relationship whereby we're performing the technical and your laboratory performing the professional and handling the billing. So that's pretty much it, John. I want to end right here on time at 1:59 Eastern, so I will turn it back to you. Thank you.
Yeah, I brought that in for a landing right on time, everybody. I appreciate that, John. I have been hearing more on the opioids being tied to relaxing the bladder, stagnant urine, causing some of these bladder cancer issues too. And so I've heard that from a few different sources. So it sounds like that might be a good market for you guys. We're going to throw up a Q&A slide. If there is any questions, feel free to ask those. But since we are up against the clock, we'll go ahead and just try to connect everybody offline, I think. So I will just put out there, if there's anything that we can do at Lighthouse for you guys, we'd love to hear about that too. So if you need help with staffing or getting a new line of testing validated inside your lab, we're doing a lot of infectious disease work right now. We've got a network of physicians in all 50 states that can help facilitate these tests. So if you need a physician order behind your test, we can do that through a telemed platform that we have. But I want to thank all the presenters, Obed, John, Sam, Jim, for your time today, your expertise. Hopefully everybody learned something. I did see there's a good amount of activity on those forums, and so we'll start connecting people offline. But I want to be respectful of everybody's time, and we'll end right here at 2. And thank you all for attending. We'll be doing a Petri Dish again next month, and we'll get that out to everybody on that topic as we develop it. Thanks, and have a great day, everyone.
Congratulations. Thank you so much, Lighthouse. Thank you. Bye-bye.
All right. Thanks, Sam.
You bet. Take care.